Hello and welcome back for more human physiology. In this video, we are going to discuss, among other things, the characteristics of water. So let's hop to it. So living things are made of both organic matter and inorganic matter. So I may have kind of indicated in a previous video that living things are made of organic matter and non-living things are made of inorganic matter. Well, that's not the case, so I apologize if I gave you that idea. So living organisms are made of both organic and inorganic matter. So how do we actually go about defining organic matter? Well, you might be surprised what the definition actually is. An organic molecule is one that contains both carbon and hydrogen atoms, and that's it. That's the only requirement. That doesn't mean that those are the only atoms those molecules will contain, but it has to contain at least those two. In living organisms, our organic molecules are likely to also contain atoms like oxygen, nitrogen, phosphorus, or sulfur, as we have indicated before. Some examples of organic molecules are fossil fuels like our gasolines and things of that nature. They are going to just contain carbon and hydrogen, so they are kind of the minimal organic molecules in nature. Carbohydrates and lipids, as we are going to study them later on in this chapter, will contain carbon, hydrogen, and oxygen. Proteins, which again we will cover later, contain carbon, hydrogen, oxygen, nitrogen, and sulfur. And then nucleic acids, the DNAs and the RNAs of the world, are going to contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So the thing that hopefully you'll notice that all four of these different classes of molecules have in common is the carbon and the hydrogen. And as we indicated, that is how we define what an organic molecule is. So inorganic molecules is going to be everything else, meaning molecules that do not contain both carbon and hydrogen. Now, that's not to say that inorganic molecules cannot contain one or the other. It just means that they don't contain both. So water, for example, H2O, contains hydrogen but not oxygen. Coal, which is basically just a lump of solid carbon, contains obviously carbon but not hydrogen. So those are not organic molecules. So for this video, we want our focus, as long as we're talking about inorganic molecules, to mostly be on water. So we have mentioned it before, water is by far the most prevalent molecule in your body. It's contained within cells, which is intracellular fluid, between cells, blood and interstitial fluid, and then as we are going to see later, it is also a participant in chemical reactions. So a couple of different functions that water provides for us. It acts as both a cushion and a lubricant, so it's a part of special extracellular fluids like synovial fluid, pleural fluid, cerebrospinal fluid, things that we will discuss later in the semester, but those are just kind of examples we can list right now. Water acts as a heat sink in the sense that probably when you discuss this in general biology, you learned that water has a very high heat capacity. So what that means is it really takes quite a lot of energy in order to either raise or lower the temperature of water, especially when you compare water to similar molecules. So this is going to be extremely important to the body's temperature homeostasis, especially as it pertains to sweating. Meaning, if sweating is all about secreting water onto the surface of our skin and then evaporating that water by using our body's own excess heat, then we can expel more heat energy because water took that much more heat in order to be evaporated. Very important here, water as a solvent, and any property for water that we discuss mostly is going to relate back to the polarity of water that we discussed in a previous video. But because of water's polarity and its ability to form hydrogen bonds, water can dissolve a very wide variety of polar or ionic compounds, so things like salt and things of that nature. So your bodily fluids, if you think about this last point of water as a solvent, your bodily fluids, whether it's intracellular fluid or extracellular fluid, those fluids are basically just water plus all of the different materials that are dissolved in the water. So when we talk about something like that, we are referring to a solution. A solution is a mixture of a liquid solvent, in this case water. Water will always be your solvent in biology, and then at least one dissolved solute. So in this picture that you're looking at right here, we have our solute, which is some type of blue crystalline material. Not sure exactly what that's supposed to be. And then we have our solvent, which is just pure water in a beaker. So if we mix those two together, we can dissolve the solute into the water to create a solution. The solution is the mixture of the solvent, which is water, and the solute, which is the material that got dissolved in there. And the whole purpose here is that this is kind of a perfect homogeneous mixture in that you cannot pick out individual solute particles away from the solvent. They basically become one big entity in and of themselves. So in biology and physiology, really, solutes can be anything from glucose to oxygen gas to sodium to potassium to chloride. Anything that is dissolved in one of our bodily fluids is considered a solute. So our definition up here, we say that a solution has to contain at least one solute. Well, if you consider our bodily fluids as solutions, they are most definitely going to contain many, many, many solutes. None of them are just going to be simple and contain one solute. They contain many, many, many. So something that we're going to have to be aware of moving forward is solute concentration. So if the solute concentration in a bodily fluid changes, meaning the number of solute particles per volume of solution, then so will the water concentration. So we want to make sure that we go ahead and kind of wrap our heads around that, especially when we get to chapter 3 and start talking about osmosis. So more on that later. So one thing we definitely want to cover right now is, as it relates to the properties of water that we've discussed, especially water's polarity, solutes themselves can either be what is called hydrophilic or hydrophobic. So the way we prefer to categorize potential solutes is on the basis of how much do they like water, how much do they want to interact with water. So if you have a hydrophilic solute, you've got a solute that really likes water and is going to dissolve and interact with water very well. So the root here, hydro, obviously means water, and phile means a lover of something. So like a bibliophile loves books, an audiophile loves music and sounds and things of that nature. So if you're a phile for something, you are a lover or an appreciator of that. So if a solute is hydrophilic, it likes water and is going to readily interact with water. So water is a polar molecule, so hydrophilic molecules are either also going to be polar or they are going to be electrically charged, either a salt or something like that. So good examples here, salts, sugars, which are polar, alcohols, which are polar. A lot of proteins of the globular type, which we'll discuss later, are polar, hydrophilic. So these are all materials, solutes, that will have no trouble dissolving in water. Now, if you've got a hydrophobic solute, then you're dealing with a type of solute that is not going to want to dissolve in water. It's not going to want to interact with water. So if you break down the roots of this word, hydro again means water, and phobia means fear of. So if you've got a phobia about something, you're scared of it. So a arachnophobe is scared of spiders. Agoraphobia is fear of open places. So the thing we want to make sure we understand here about a phobia, so if you're an arachnophobe, you're afraid of spiders, well, that implies something extra. Not only are you afraid of spiders, but you are going to do everything in your power to make sure that you never come across spiders. So you're going to take all kinds of precautions to make sure that you never see a spider. That's the way you're going to live your life happiest. So what that means for a hydrophobic molecule, not only does it not like water, it is going to avoid water at all costs. And this is going to be a particularly important topic for us when we get to chapter 3 and we start talking about the cell membrane. So we'll just want to go ahead and file this away for later. A hydrophobic molecule does not like water and will stay away from water. So some examples here are nonpolar gases like oxygen and carbon dioxide. Fat molecules are very nonpolar, so they're not going to like interacting with water. Same goes for steroid. A lot of steroid hormones like testosterone and estrogen. Fibrous proteins like collagen and keratin. So hydrophobic molecules tend to be very nonpolar. So a kind of a general good maxim in biology is that like goes with like. Polar molecules like to interact with other polar molecules. Nonpolar molecules like to interact with other nonpolar molecules. But polar and nonpolar molecules do not like each other. It's for this reason that if you've ever, say, made salad dressing, anytime you want to use it, you got to really shake up the bottle well, because the salad dressing contains both water and oil. And the oil, which is nonpolar, will not want to mix well with the water, which is polar. So you kind of have to force them together by shaking the bottle up. Okay, so let's talk about that issue of solute concentration. So to kind of give you an idea of what we're looking at here, we are looking at 4 different test tubes. So obviously you can tell that the volume of each is about the same. So we're saying there's about 100 milliliters of water in each tube. And obviously you can tell the main thing that's different here is the color of each. Well, so what I am telling you here is that each one of these test tubes contains 100 milliliters of water, and we have dissolved either 10 grams, 5 grams, 2 grams, or 1 gram of a red solute in here. So imagine kind of a red crystalline powder that we have dissolved in the water. So if we are calculating concentration, concentration is just mass of the solute divided by the volume of the solution. So the first concentration of the leftmost tube would be 10 grams divided by 1/10 of a liter, which is what 100 milliliter is. So the concentration here is 100 grams per liter. Well, the next tube, since it has half as much solute, the concentration is going to be half as much, since the volume of the two are the same, so 50 grams per liter. The next tube is going to be 20 grams per liter, and the last tube will be 10 grams per liter. So we've got 4 different tubes here, and we can tell not only based on the mass of the solute that we've added in there, but also on the color of the solution, how deep the color is, we can tell that the concentrations of the 4 tubes are very different. Now, we are looking at this in terms of solute concentration, so we can tell the tube on the left has the greatest solute concentration, and the tube on the right has the lowest solute concentration. But a skill that we want to go ahead and start developing is the ability to also look at this in terms of water concentration. So we can tell that the tube on the far left has the greatest solute concentration. Does it also have the highest water concentration? Well, the thing that you're going to figure out before too much longer is that the higher the solute concentration, the lower the water concentration. So imagine that we've got 100 milliliters of solution. So we've got kind of a finite space for all the molecules, whether it's water molecules or solute particles. So the more solute particles, the less space is available for water, and the concentration of the water itself will be lower. So even though the tube on the far left has the greatest solute concentration, it has the lowest water concentration. And then conversely, the tube on the far right, it has the lowest solute concentration, but the highest water concentration. So solutions are just one type of liquid mixture. It's certainly not the only type of mixture that we will encounter throughout the semester. So we can also deal with things like colloids and suspensions as well. So colloids are kind of like solutions, except that the solute particles are not going to completely dissolve and become kind of a clear solution. So the solute particles are particularly dense, and they are going to scatter light, meaning that unlike, say, this solution in which it is a clear solution that you can see through, a colloid solution is going to be completely opaque. Milk is a very prominent example. Blood is a perfect example of a suspension. Now, the blood plasma, which is only the liquid solution portion of the blood, that is actually a solution. That is actually water as a solvent, including a lot of different salts and proteins and stuff dissolved in. What makes whole blood a solution is that you've got a number of different cells, like red blood cells, white blood cells, and platelets, that do not dissolve in the water, and if you leave a sample of blood sitting out, those things will settle out of solution over time, meaning they would sink to the bottom of the test tube. So the hematocrit is a way of actually taking advantage of this to measure the percentage of whole blood that is actually taken up by red blood cells. And that's kind of important since red blood cells carry oxygen around. It gives you kind of a good idea of, for a person, to what extent are they capable of transporting oxygen throughout your body. And then a related test called a sed rate test measures the amount of time it takes for red blood cells to actually settle to the bottom of the tube on their own. So one of the last things that we want to talk about here as it pertains to water itself is that water, in addition to being a component of bodily fluids, acting in temperature, homeostasis, and things of that nature, water is also a willing and able participant in chemical reactions, especially in cases in which we are building and deconstructing very large molecules, our catabolic and anabolic reactions. So in this way, water can participate in one of two different types of reactions. The first is called a dehydration synthesis. So dehydration synthesis involves the liberation of one molecule of water in order to form a covalent bond between two compatible molecules. These two compatible molecules are what we call monomers. So in the picture here on the top, we have monomer 1 and monomer 2. You'll notice that each monomer has an OH group attached to it, and if you take one oxygen and two of the hydrogens, we actually can see a water molecule that is ready to be liberated. So what a dehydration synthesis does is it forms a chemical bond in between these two monomers, and it liberates that OH and H, leaving the other O behind as kind of the part of the chemical bond there, and we liberate a molecule of water. So dehydration synthesis is all about forging covalent bonds in between two smaller molecules. So we are starting with two small molecules, forming a chemical bond, and building a bigger molecule. So something for you to think about, would that be an anabolic reaction or a catabolic one? The next type of reaction is called a hydrolysis reaction, and that is the one down here at the bottom. The hydrolysis reaction is essentially the dehydration synthesis run in reverse. So with a hydrolysis reaction, we already have a fairly complex molecule that is joined by a covalent bond, and we are utilizing a free water molecule in order to break that chemical bond. So we use the water molecule to break the chemical bond, and now you can see the remnants of the water molecule contained within monomer 1 and monomer 2. So in this case, we are starting with a complex molecule, breaking a chemical bond to create simpler molecules, smaller molecules. Would that be anabolic or would that be catabolic? So regardless of which one of these reactions that we're talking about, whether we are talking about building chemical bonds by using dehydration synthesis or breaking chemical bonds by using hydrolysis, keep in mind that these are chemical reactions that have to be catalyzed by particular enzymes. So these are not the sorts of reactions that are just going to happen all by themselves willy-nilly. They will only happen when an enzyme is active and present and willing and able to catalyze the reaction, just like we talked about before. So something that we mentioned before, kind of going back to the whole hydrophobic, hydrophilic aspect, we talked about how salts, ionic compounds like sodium chloride or potassium iodide, are readily dissolvable in water. So in table salt, for example, the sodium chloride that we looked at, when that salt is a solid and not yet dissolved in water, it is electrostatic interactions that hold the positive sodiums and the negative chloride ions together. So we already mentioned that water is able to very easily break these bonds and solvate the sodium and chloride ions, meaning that if you look over here, when you dissolve salt in water, you're going to have just individual sodium ions that are surrounded by water and individual chloride ions which are surrounded by water. So if you are to dissolve a salt like sodium chloride in water and you start thinking about your solution, you have two different solutes. You've got sodium ions and you've got chloride ions. So if you're thinking about concentrations for those solutes, you would have to consider separately the concentration of sodium, and the concentration of chloride. They are two completely separate things. So a good rule of thumb, salts are always going to completely dissociate in water. So meaning when you add sodium chloride to water, there is no longer sodium chloride. It's no longer together. So you have individual sodium and chloride ions. So again, as I mentioned before, this is going to be critical later in the semester because of the electrical charge of these ions. This is going to allow for electrical currents in the case of things like nervous system signaling. So another surprising thing, maybe not if you've already taken your chemistry class, which I think all of you have, water itself can dissociate into ions. It does so according to this equation here. You start with a molecule of water, and it breaks up into a hydrogen ion and a hydroxide ion, or OH. Now, thankfully, water does not do this very often. It only does this about 1 in every 10 million water molecules. So the concentrations of those hydrogen ions and hydroxide ions in pure water are each going to be 1 × 10 to the -7th molar, or moles per liter. So not very concentrated. So when we start talking about acids and bases, these are going to be substances that when added to water have the ability to change the concentration of both hydrogen ions and hydroxide ions. So an acid here on top, an acid is a substance that when you add it to water, it is going to increase the concentration of hydrogen ions, whereas a base is going to do the opposite. A base will cause the hydrogen ion concentration to decrease, or alternatively, you could think of it as the hydroxide ion concentration increasing. So this is all important because we have something called the pH scale. We have for aqueous solutions like pure water or like salt water or blood or any of our bodily fluids, we have the ability to measure how much hydrogen ion is present. And that is what the pH scale is all about. So pH can be calculated by using this formula here, the negative log base 10 of the hydrogen ion concentration. Now, before you start having kind of Vietnam flashbacks back to chemistry, don't worry, we're not going to be doing any of those calculations in this class. But we do need to kind of appreciate what this equation is kind of telling us. So this is a logarithmic scale on the basis of units of 10, kind of like the metric system. So because every unit on the pH scale is off by a difference of 10, this is going to mean, for example, that if we have something like your blood, which is at a pH of 7.4, and we compare it to urine, which has a pH of 6.4, because there is a difference of exactly 1 pH unit between them, blood is 10 times more basic than urine, or alternatively, you could say urine is 10 times more acidic than blood. So the idea here is that 7 on the pH scale ranges from 0 to 14, a 7 on the pH scale is perfectly neutral, and that is pure water. Pure water is perfectly neutral. But when you start adding things to water, you can start changing the pH a little bit. So we want to be aware, therefore, that because all of our bodily fluids are aqueous in nature, they have different solutes contained within the water, all of our bodily fluids are going to have associated pH values. And that is going to be important because it's just another thing that we want to maintain homeostasis for. So consider this. Because your metabolism is the sum of thousands upon thousands upon thousands of different chemical reactions that are all releasing products and metabolites and things of that nature, those metabolites may be able to act as acids or bases. So, for example, cellular respiration. Every time your cells perform cellular respiration, they release carbon dioxide. Well, carbon dioxide can partially dissolve in the blood and change the pH of the blood, so that can be kind of a problem for us. So your metabolic reactions can, over time, cause the pH of your bodily fluids to change. Well, if we know anything about homeostasis by now, we don't want that. We don't want things to change. So your body's homeostatic defense mechanism against pH changes is buffers. So a buffer is a mixture of chemicals that will steady the pH by absorbing hydrogen and hydroxide ions as needed, so we don't get really big changes in the pH that ordinarily would happen. So one very common buffer mechanism that we will probably not discuss again until the respiration chapter much later in the semester is carbonic acid and bicarbonate. So both of these, both carbonic acid and bicarbonate ions, are present in the blood, and they are capable of absorbing any hydrogen and hydroxide ions that are produced so that we can maintain a homeostatic blood pH of 7.4, even though the constant performance of cellular respiration is always trying to make our blood more acidic. Having this buffer system in place allows our blood pH to stay the same, even though it's always trying to change. All right, so that is going to do it for this particular video. I thank you for your attention. Next time, we are going to start focusing on organic molecules, particularly the carbon backbone of a lot of our organic molecules like carbs and lipids and proteins and nucleic acids, and the types of functional groups that they can contain, and how those functional groups can impact the actual chemical nature of those molecules. So thank you very much, and